Have you ever stopped to think about what's really happening inside your body after you eat? I'm not just talking about digestion. I mean a deep chemical conversation between your food, the trillions of microbes in your gut, and your very own DNA. Well, today we're going to dive into the science of transcriptomics and actually eavesdrop on that conversation.
Now that might sound like something out of a sci-fi movie, but I promise you, it's very real. We're going to explore the incredible technology that lets scientists answer that exact question, revealing how your body's genetic blueprint is being actively rewritten moment by moment based on how you live your life.
So to get our heads around this, we need to look beyond the static DNA that's locked away in our cells. We need to focus on the active, working copy of our genetic instructions, the stuff that's happening right now.
Think of it like this. Your DNA is like a massive library containing every single instruction manual your body could ever possibly need. The transcriptome, though, that's the list of manuals that are actually checked out and open on the workbench right now. It's a dynamic, live snapshot of what a cell is up to, and it's constantly changing based on things like, you guessed it, the food you just ate.
But here's the catch. When we try to read this active blueprint, we run into a huge problem. Most of what we extract from a cell isn't the valuable messages we're looking for. It's basically just noise. So how in the world do you filter out all that static?
And that brings us to the first crucial step, which is a classic needle in a haystack problem. But, lucky for us, scientists have figured out a really clever way to solve it. And I do mean a gigantic haystack. Get this, in a typical RNA sample, over 95% of it is something called ribosomal RNA, or rRNA. Now, it's essential for building proteins, which is great, but for our purposes, it's just static that completely drowns out the messenger RNA, the mRNA, which carries the actual instructions we want to read. So how do you do it? With a brilliant technique called the ribo-zero method. It's basically a microscopic fishing trip. You introduce these tiny little probes with magnetic beads on them. Think of them like tiny fishing hooks designed to catch only one type of fish, that noisy rRNA. They latch on, ignoring everything else. Then you bring in a magnet, and poof! Just like that, the magnet pulls the entire rRNA haystack right out, leaving behind only the pure, valuable mRNA messages. A perfectly clean sample.
Okay, so we've found our needles, we've successfully isolated the messages, but now comes the next challenge, actually reading what they say. So we've got our pure mRNA, but reading it isn't as simple as just opening a book. First, we actually have to shred it into millions of tiny pieces, creating a massive biological puzzle. This whole process is called next-generation sequencing, and while it looks complex, the idea is pretty simple. To read a really long mRNA message, the machine first shatters it into millions of tiny overlapping fragments that we call reads. So now we have the text of all these little pieces, but we've completely lost the original order. It's kind of like taking 1,000 different books, putting them all through a shredder at once, and then trying to put every single one back together perfectly. I mean, how could you possibly do that? The answer, it turns out, is bioinformatics. We use a reference genome as our picture on the box. We have this powerful software that takes every single one of those millions of short reads as they come pouring in, and it finds its one unique home on that reference map. It's a huge digital matching game where you get to see every single puzzle piece just click perfectly into place. And here is the magic moment. Once all the puzzle pieces are mapped, we just count them. If thousands and thousands of reads land on a specific gene, like gene A right here, they stack up into this huge mountain of data. That tells us that gene was highly active or upregulated. If only a few reads map to another gene, like gene C, it's just a tiny hill. The height of that mountain is the data. That's how we turn a biological sample into a precise measurement. Okay, so that's the technology, and it's incredible. But what can it actually tell us? Let's go back to our original question and look at a fascinating real-world experiment. In one study, scientists gave a group of mice a standard diet, and another group got a single meal that was designed to be like human fast food. Then they just waited for only 4 hours and asked a really simple question. Did anything change? And they used transcriptomics to find out. So the very first changes happened in the gut, and they happened incredibly fast. The fast food meal immediately shifted the whole community of gut microbes. Specifically, the levels of important bacteria like Turricidobacter just plummeted. And when the bacterial population changes, that immediately changes the cocktail of chemical signals they're all pumping out.
Now, these altered chemical messages didn't just stay in the gut. They were absorbed into the bloodstream and acted like a message in a bottle sent from the gut microbiome with a very specific destination, the liver.
And here it is, the stunning result. This heat map is a snapshot of the liver's transcriptome,
Each column is one mouse, and each row is a single gene. Red means that gene's activity is turned way up, blue means it's turned way down. Now, just look at the two groups. You don't need to be a scientist to see the massive difference between the mice on the normal diet on the left and the fast food group on the right. The liver got the message all right, and it responded by completely rewriting its genetic program.
I mean, this is a profound discovery. The fast food wasn't just energy, it was information. It sent a signal through the gut microbes that told the liver to fundamentally change its entire metabolic operating system.
So, what's the big takeaway from this incredible biological story? Let's put it all together. What we're seeing is this astonishingly fast chain reaction. In just 4 hours, a single meal sets off a domino effect that travels from your plate to your gut bacteria and ends with the total reprogramming of your organ's genes. And this isn't just a theory. The researchers themselves concluded, and I'm quoting here, that a single meal can have profound effects on microbiome community structure and rapid transcriptional reprogramming of the liver. It just highlights this powerful, immediate connection between our diet and our genes. And ultimately, that is the power of transcriptomics. It's a revolutionary tool that lets us eavesdrop on the constant, invisible chemical conversations happening inside of us, revealing a level of biological complexity we never even knew existed.
And that just leaves us with a final fascinating question. If a single meal can do all of this, what other secret conversations are shaping our health every single moment? What messages are being sent and received inside you right now?